Hello everyone, welcome back to the lecture series of overview and integration of cellular metabolism. This is lecture class 30 where we are going to discuss about cholesterol metabolism. So, in this class what we are going to discuss is about biosynthesis of cholesterol, then how this cholesterol synthesis is regulated, different compounds which are formed from cholesterol, what is the fate of cholesterol, and finally few relevant clinical or applied significance with respect to cholesterol metabolism. So, before going into the metabolism or biosynthesis of cholesterol, let us discuss a bit biochemistry of cholesterol. That is, cholesterol is, as you can see in the structure, that this is a 27 carbon compound. It contains a sterol ring in it. So, this is our steroid nucleus or sterol ring. Sometimes we call it CPP ring, cyclopentanoparhydrophenanthrin ring. So, this is not a chemistry class. So, I am not going into much detail of this. Then what is the importance of cholesterol metabolism? I mean, why we are very much intrigued with cholesterol metabolism. Now, cholesterol, remember, even if we very frequently say the cholesterol is increased, cholesterol is bad, cholesterol should be reduced, etc., but remember, cholesterol is one very important and required product in our body and for our regular maintenance of human body actually. Why? Because one very important thing, cellular membrane. So, cholesterol is one important integral part of cell membrane. Then it is also required for nerve conduction because it imparts an insulation effect over the nerve. Then it has some biosynthetic property. It acts as precursor for compounds like bile acid and bile salt. Then steroid hormones, all the steroid hormones are actually synthesized from cholesterol, starting from glucocorticoids, all the glucocorticoids, then androgens, estrogens, they all are synthesized from cholesterol, and one vitamin, very important, vitamin D3, esterification of vitamin B3, for that cholesterol is required. So, these are the important compounds or important functions of cholesterol for which cholesterol synthesis is very intriguing metabolic topic we need to know. So, let us move to the synthesis of cholesterol. Now, synthesis of cholesterol occurs almost in all the tissues, but the most important one is liver, then intestine, skin, adrenal cortex, and in reproductive tissue. Every day, daily around 1 gram of cholesterol is synthesized in adult cells. Then the synthesizing enzymes are present in cytosol as well as microsome. So, again the biosynthesis of cholesterols occurs in with the help of cytosolic as well as microsomal enzyme. What is the precursor for cholesterol synthesis? Is acetyl coenzyme. Remember, acetyl coenzyme is the precursor for all the carbons in cholesterol. 27 carbons, they all are actually derived from acetyl coenzyme. And then one reducing equivalent is required, that is NADPH, and also the energy is provided by ATP. So, these are the basic points you need to remember for biosynthesis of cholesterol. Now, cholesterol synthesis is one complex mechanism, actually complex series of steps which finally forms cholesterol, but we have divided the cholesterol synthesis in 4 important stages or phases rather. So, the first phase or stage is formation of mevalonate. Formation of mevalonate from 3 acetyl coenzyme or acetate units. Then that mevalonate is finally forming isoprene unit or activated isoprene unit in stage 2. Now, isoprene units are 5 carbons. So, remember mevalonate was 6 carbon compound. From that 1 carbon is actually removed to form the 5 carbon isoprene unit. That isoprene units are actually condensed to form squalene, that is a 30 carbon compound, cholesterol is 27 carbon compound. So, finally, and also cholesterol is one cyclical structure. I told you there is a ring, sterol ring or CPP ring is there, but all this up to squalene, these are all linear compounds. So, what we need is cyclization of the squalene to form the rings of sterol rings or steroid nucleus, and there are further series of changes even after that sterol nucleus formation, that is oxidation, removal or migration of methyl groups to finally form cholesterol, which is the 27 carbon compound, and in between that the ring structure is first formed in lanosterol. So, these are the 4 stages in biosynthesis of cholesterol. Now, we will discuss these stages and what are their specific metabolic steps in under these phases. So, this is the overview that acetyl coenzyme A is a 2 carbon compound. So, they are forming mevalonate via HMG coenzyme A. We will discuss HMG coenzyme A. Then mevalonate is the 6 carbon compound. From 6 carbon compound there is formation of isoprene unit. Isoprene units are 5 carbon compounds. These 5 carbon compounds are actually condensed to form 2 such 5 carbon compounds are condensed to form the activated 10 carbon compound. Then another 5 carbon compound is added to the 10 carbon compound to form C15 compound. Finally, these 2 C15s are condensed to form linear squalene. Then there is cyclization to form lanosterol. Now, in this lanosterol, this is the first structure of in the pathway of cholesterol synthesis. This is the first cyclical structure, and then further modification to form cholesterol. So, this is the gist of cholesterol synthesis, fine. So, let us see one by one what happens, that from acetyl coenzyme A, first which is formed is acetoacetyl coenzyme A. So, there is condensation of 2 molecules of acetyl coenzyme A with the help of the enzyme acetoacetyl coenzyme A synthase. Remember, this is synthase, and synthetase are those enzymes where ATP is required. In synthase, ATP is not required. So, here it is synthase, acetoacetyl coenzyme A synthase, sometimes we call it thiolase as well. So, there is formation of acetoacetyl coenzyme A from 2 molecules of acetyl coenzyme A. Then there is formation of HMG coenzyme A, that is beta hydroxy H beta methyl glutaryl coenzyme A. So, this is our HMG coenzyme A, and HMG coenzyme A is formed by condensing another acetyl coenzyme A to acetoacetyl coenzyme A. So, this is our 6 carbon compound, and of course, there is a release of coenzyme A because here was one coenzyme A, another coenzyme A was there. So, one coenzyme A molecule is released, and then HMG CoA further undergoes reduction to form mevalonate. Now, the reducing equivalent as I told you here is NADPH, remember this is NADPH, and also the coenzyme A is removed to form mevalonate. Once again, mevalonate is 6 carbon compound. Then from this mevalonate, isoprene units are formed. Now, isoprene units are 5 carbon compounds, so definitely there must be one decarboxylation, where carbon dioxide is released, one carbon dioxide is released. Now, what happens during the formation of isoprene unit from mevalonate, there is phosphorylation with ATPs, and this phosphorylation happens for 3 times. So, here you can see the first phosphorylation, second phosphorylation and third phosphorylation. Along with that there is, and the last phosphorylation is followed by decarboxylation. So, what is formed? For the first intermediate is 3-phosphopyrophosphomevalonate. After 3 phosphorylation, what we are getting is 3-phospho-5-pyrophosphomevalonate, and here you can see one kinase is there, one phosphotransferase is there, and one decarboxylase is there. Fine. So, what we are getting after decarboxylation is the first isoprene unit, that is isopentenyl pyrophosphate. Isopentenyl pyrophosphate. So, once again mevalonate undergoes 3 steps of phosphorylation with 3 molecules of ATP and also decarboxylation to form the first isoprene unit, which is isopentenyl pyrophosphate. Now, there are 2 types of isoprenoid units are formed. One is isopentenyl pyrophosphate and its isomers. So, next step is isomerization of isopentenyl pyrophosphate to form dimethyl allyl pyrophosphate. This is another isoprene unit. So, from mevalonate we are getting 2 isoprene units, one is isopentenyl pyrophosphate, another is dimethyl allyl pyrophosphate, and both of them are required in cholesterol synthesis. So, after formation of this isoprene unit, what will be there is condensation of this 5 carbon isoprene unit. So, first is formation of the 10 carbon compound geranyl pyrophosphate. Now, what happens? There is condensation of one molecule of dimethyl allyl pyrophosphate and isopentenyl pyrophosphate in head to tail fashion. Now, head group is the part where pyrophosphate is attached. So, the head group of dimethyl allyl pyrophosphate and the tail part of isopentenyl pyrophosphate are condensed to form geranyl pyrophosphate, which is a 10 carbon compound, and definitely here you can see the pyrophosphate is released. Next, there is another condensation. This geranyl pyrophosphate is once again condensed with another molecule of isopentenyl pyrophosphate in head to tail fashion, head of geranyl pyrophosphate containing the pyrophosphate part and tail of isopentenyl pyrophosphate is forming 15 carbon compound, that is farnesyl pyrophosphate. Farnesyl pyrophosphate is a 15 carbon compound. So, what we are seeing here are 2 5 carbon compounds which are condensed to form the 10 carbon compounds geranyl pyrophosphate. Geranyl pyrophosphate is once again condensed with isopentenyl pyrophosphate to form the 15 carbon compound farnesyl pyrophosphate. Now, these farnesyl pyrophosphate, 2 such molecules are condensed to form 30 carbon compound squalene, and this condensation occurs in head to head condensation. So, basically what happens, the both the pyrophosphate containing ends are actually condensed. So, you can see from both the ends 2 pyrophosphates are released, and this is one NADPH dependent reaction where the enzyme is squalene synthase. So, what we are getting after condensation of the 5 carbon units is one 30 carbon compound squalene, and this squalene, remember this squalene is the linear structure. Now, squalene synthase, what you need to remember, need some cofactors like magnesium, manganese and cobalt, and squalene synthase is one microsomal or smooth endoplasmic reticulum attached enzyme. So, at the end of the condensation of these 5 carbon isoprene units, what we are getting is squalene. Squalene is one 30 carbon compound, and remember this is a linear structure. So, what we need is cyclization to form the sterol ring. So, next we are going to discuss about cyclization. So, squalene undergoes cyclization with the help of one monooxygenase, which is known as squalene monooxygenase, and this squalene monooxygenase once again is one NADPH dependent enzyme. Now, you can see here molecular oxygen is required. Now, that molecular oxygen, one atom is attached to, you can see at the end of the squalene structure, forming squalene 2,3 epoxide, and another atom of oxygen is actually forming water. Here you can see is utilized to form water. So, squalene epoxide then undergoes cyclization by cyclase enzyme, which forms a 30 carbon ring structure. So, this is the first ring structure, that is known as lanosterol. Lanosterol, remember once again, this is one very important question, what is the first ring structure which is formed during the synthesis of cholesterol? That is lanosterol, and this is the first steroid compound. Now, lanosterol undergoes around 20, approximate 20 steps of reaction to form cholesterol. Now, all the steps are not till date even not explained in details, but there are few important steps which have been discussed vividly. So, we are going to just highlight those structures. So, from lanosterol, after multiple steps we are getting zymosterol, from zymosterol we are getting desmosterol, and finally this desmosterol is forming cholesterol. Now, these modifications you might not remember, but for the information I will say that there are modifications like, as I told, methyl group addition in lanosterol leads to zymosterol formation. Then there is transfer or migration of double bonds in zymosterol to form desmosterol, and finally those the desmosterols, there those double bonds are actually reduced by NADPH to form cholesterol. So, this is how cholesterol is formed, but remember actually in between there are around 20 steps. So, these are the important steps I have highlighted from lanosterol to cholesterol formation. So, once again to recapitulate that the precursor molecule is acetyl coenzyme A. Three such acetyl coenzyme is condensed to form the 6 carbon compound mevalonate. Mevalonate forms the activated isoprene unit, that is isopentenyl pyrophosphate, as well as its isomer dimethylallyl pyrophosphate. These two isoprene units are condensed to form 10 carbon compound geranyl pyrophosphate, then 15 carbon compound farnesyl pyrophosphate, and finally squalene 30 carbon compound, and this 30 carbon linear compound is actually undergoes cyclization to form lanosterol, and lanosterol after around 20 steps form cholesterol. Now, one very interesting thing about squalene is squalene was actually derived from sharks. So, the name came from the squalus term, and Then also these compounds like farnesyl pyrophosphate, geranyl pyrophosphate, these isoprene units are actually the naturally occurring perfumes or aroma generating material. So, like geranyl was actually derived, the name is derived from geraniol, which is present in rose oils. Similarly, farnesyl term is actually derived from Farnesia acacia plant, that is also one aroma generating plant. So, these are the names are generated from, names are given from where these compounds are actually initially derived. So, this is all about biosynthesis of cholesterol. Now, we are going to discuss the regulation of cholesterol. Now, remember once again, regulation of cholesterol, it has few long term regulation where we will discuss the synthesis of different enzyme, or I mean we are dealing with the numbers of enzymes, as well as few short term regulation where we are talking about, we will be talking about activation or deactivation of enzyme. Now, cholesterol synthesis of cholesterol is regulated by hormones. Hormones give signals for the regulation of cholesterol, as well as the intracellular cholesterol content is one very important decisive factor for cholesterol synthesis. Now, the rate limiting step is the formation of mevalonate from HMG coenzyme A, and the enzyme which is regulated or the regulatory enzyme in cholesterol synthesis is HMG coenzyme A reductase. Now, let us see how this long term regulation happens. So, long term regulation, here we are talking about the synthesis, decreased or increased synthesis of the regulatory enzyme, that is HMG coenzyme A reductase. Now, remember transcriptional control is for cholesterol synthesis is not only for HMG coenzyme A reductase, but other enzymes are also regulated in this transcriptional control. The most important one is HMG coenzyme A reductase, obviously. So, to induce or regulate the transcription, what we need is one transcription factor through which most of the long term regulation or transcriptional control imparted to the enzymes. Here the transcription factor is SREBP. Remember, we already have discussed SREBP. Now, we are going to discuss in details how this SREBP is controlled by the intracellular cholesterol. Now, SREBP stands for sterol regulatory element binding protein. In many books or journals, it is also represented as sterol responsive element binding protein. And another adjunct protein is there, that is SCAP protein. The it is the full form of SCAP is SREBP cleavage activating protein. So, SCAP protein is another adjunct protein. Now, the transcription factor is SREBP. Now, this transcription factor SREBP is a protein definitely. Now, its amino terminal is the soluble part, and this amino terminal imparts the actual transcriptional regulation activity. But the problem is this amino terminal is not free. So, the whole SREBP molecule is actually attached to the membrane of endoplasmic reticulum. So, here you can see SREBP, here it is attached with the endoplasmic reticulum. So, it is not freely movable, or it just cannot enters nucleus, cannot bind with the DNA as it is. What is needed is one regulatory signal. Now, remember, when there is adequate amount of cholesterol present in cell, it is sensed by the SCAP protein. So, SCAP protein is actual sterol or cholesterol sensor here, and this SCAP protein is attached with the SREBP. So, SREBP and SCAP protein is they are attached together and is attached to the endoplasmic reticulum membrane. Now, when there is reduced cholesterol content in cell, it is sensed by the SCAP protein. After sensing the reduced cholesterol content, what happens are two proteolytic cleavage. Now, in first proteolytic cleavage, this SCAP protein is separated from the SREBP. So, here you can see the SREBP is free from SCAP protein, but it is still attached to the endoplasmic reticulum. The second proteolytic cleavage, it breaks the amino terminal, rather it detaches the amino terminal segment of SREBP from the membrane bound fraction. So, now the amino terminal of SREBP is actually free to migrate inside the nucleus. Inside the nucleus, it binds with the respective genes, and those genes are actually of HMG coenzyme reductase synthesis or other cholesterol metabolizing enzymes related genes. So, now the synthesis is increased when there is cholesterol, the cellular content of cholesterol is low. So, once again the low concentration of cholesterol is sensed by SCAP protein. SCAP protein, on sensing the cholesterol low content, it undergoes two proteolytic cleavage to release the amino terminal domain of SREBP. That amino terminal domain enters nucleus, binds with the respective genes and causes increased synthesis of HMG coenzyme reductase, which increases the synthesis of cholesterol. Now, when by this induction, when the cholesterol content is finally increasing, that is also sensed by the SCAP protein. SCAP protein again reattaches with the SREBP fragment, and SREBP now is not able to move inside the nucleus. So, the synthesis is now decreased. So, this is on demand cholesterol based on the available cholesterol concentration inside the cell. Now, HMG coenzyme reductase enzyme itself can be itself can sense the signal of low or high cholesterol content. Now, HMG coenzyme reductase enzyme itself is one integral membrane protein, and it is attached to the endoplasmic reticulum. As I told, it is the microsomal enzyme. So, this enzyme, its cytosolic domain is acting as the enzyme. So, this is imparting the catalytic effect, whereas the membrane domain, this is our membrane domain, it acts as the cholesterol sensor. The sensor is just like the SCAP protein, it has the similar domain which is present on the SCAP protein. So, basically what happens when there is high cholesterol content inside the cell, it is sensed by the membrane bound domain of the HMG coenzyme reductase. On sensing, there is conformational change in the enzyme HMG coenzyme reductase. Such conformational changes makes this enzyme vulnerable for proteolytic degradation. So, HMG coenzyme reductase is actually proteolytically degraded. So, cholesterol synthesis is decreased. So, this is how HMG coenzyme reductase itself can sense the cholesterol content of the cell. Then there is short term regulation, whereby where the enzymes are activated or deactivated following covalent modification. Here the covalent modification is following phosphorylation dephosphorylation cycle. The HMG coenzyme reductase enzyme is active when it is dephosphorylated and inactive when it is phosphorylated, and this phosphorylation dephosphorylation is triggered by hormones. So, cortisol or glucagon, they causes the phosphorylation. So, basically they are decreasing the HMG coenzyme reductase activity. So, basically they are decreasing the cholesterol synthesis. Similarly, when there is excessive exercise, that exercise there is breakdown of ATP. ATP is the breakdown product of ATP is AMP. We already have discussed AMP acts as an activator of AMP kinase. This AMP kinase once again phosphorylate HMG coenzyme reductase, and that causes inactivation of the enzyme. On the contrary, insulin or thyroxine, they activates HMG coenzyme reductase by activating the phosphatase or dephosphorylation and increases the synthesis of cholesterol. So, what we can see in regulation of cholesterol synthesis, important hormones are there, insulin and glucagon, as well as the intracellular cholesterol content is also acting as a signal for cholesterol synthesis, as well as receptor mediated uptake of cholesterol from the circulation. So, what happens if there is excess amount of cholesterol in circulation, it forms if there is excess amount of cholesterol in cell, it forms oxysterol. Now, oxysterol inhibit the receptor mediated endocytosis of cholesterol. Similarly, if there is excess amount of cholesterol in cell, it activates the ACAT enzyme, acetyl coenzyme acyl transferase, or acyl coenzyme acyl transferase, that forms cholesterol esters. Similarly, AMPK or glucagon or cortisol, they cause inactivation of HMG coenzyme reductase, or insulin activates, insulin and thyroxine, they activates the HMG coenzyme reductase. So, these are the nutshell of regulation of cholesterol synthesis. Then the clinical or applied importance is there are multiple drugs which helps in lowering the intracellular cholesterol synthesis by inhibiting the enzyme HMG coenzyme reductase. Those drugs are statin group of drugs. The compounds are like lovastatin, simvastatin, like that. So, these are the statin group of drugs. Now, statin group of drugs acts as competitive inhibitor of HMG coenzyme reductase. Basically, lovastatin in inactive form, it is one lactone. Now, when ingested, it is hydrolyzed to form the active form, that is a beta hydroxy derivative of lovastatin. This beta hydroxy derivative is actually competitively inhibiting HMG coenzyme reductase, thereby decreasing the intracellular synthesis of cholesterol. So, remember, statin group of drugs are cholesterol lowering drugs. Then the fate of cholesterol in our body. Now, cholesterol is supplied in our body either by diet, or that is exogenous cholesterol, or endogenous cholesterol, which are circulating inside the body or synthesized in our body, then de novo synthesis which is occurring in liver. So, these are the the sources of cholesterol in our body. What are the fates of cholesterol in body? Cholesterol is secreted in the form of VLDL. So, this is our circulatory form of cholesterol. So, cholesterol is secreted from liver as VLDL. It can be secreted as free cholesterol and is liberated in bile, or the liver cholesterol can be converted to form bile acid or bile salts. Now, remember, the free cholesterol which is released in bile, it can be it is actually excreted through feces. Now, this amount is around 1 gram per day, and there is a bit of ring modification which is finally forming coprostenol or cholestanol, and that is done by colonic bacterial activity. A part of this free cholesterol which is secreted in bile, it can be absorbed in intestine and can be exported as chylomicron. So, this is the fate of free cholesterol in bile. Remember, cholesterol can be forming bile or cholesterol can be secreted as it is in bile. So, here we are talking about the free cholesterol. Now, the part of bile synthesis of cholesterol, a bit we need to know about that. The bile is synthesized in liver cell definitely, and there are few primary bile acids, like two primary bile acids, cholic acid and chenodeoxycholic acid. Then secondary bile acids like deoxycholic acid and lithocholic acid. A bit of the bile acid synthesis I am going to discuss. So, this is formation of bile acid. Now, here you can see cholesterol undergoes hydroxylation in the 7th position of the cholesterol link, 7 alpha hydroxylation, and the enzyme is 7 alpha hydroxylase to form 7 alpha hydroxycholesterol. Then 7 alpha hydroxycholesterol without hydroxylation can form chenodeoxycholic acid, or after hydroxylation in the 12th position can form cholic acid. So, again the two primary bile acids are formed from 7 alpha hydroxycholesterol. Cholic acid is formed after 12 alpha hydroxylation, which is an NADPH dependent enzyme. Then there is release of a 3 carbon compound in the form of propionyl coenzyme A that forms cholyl coenzyme A finally, and without 12 alpha hydroxylation, here you can see there is no 12 hydroxylation, no another hydroxylation is not there, but there is release of a 3 carbon compound propionyl coenzyme A. Now, here you can see these 3 carbons are released as propionate, then there is oxidation at the 24th carbon, also 7 alpha hydroxylation as well as reduction of the double bonds. These are common for all the primary bile acid. Along with that, one additional step is there for the cholic acid, that is 12 hydroxylation, 12 alpha hydroxylation. So, what we are getting, two primary bile acids, that is cholic acid and chenodeoxycholic acid, they are conjugated with taurine and glycine to form taurocholic acid and glycocholic acid, or similarly taurochenodeoxycholic acid or glycochenodeoxycholic acid. So, these are the primary bile acids, and their salt version are sodium or potassium taurocholate, glycocholate, taurochenodeoxycholate or glycochenodeoxycholate. So, these are the primary bile acids. Now, primary bile acids, when they are treated with colonic bacteria, they undergoes deconjugation and also dehydroxylation to form the secondary bile acids. Those are deoxycholic acid and lithocholic acid. So, these are our primary bile acid and secondary bile acid formation. Now, remember, bile salts keeps recycling in our body. So, liver only synthesizes 0.2 to 0.6 gram per day bile salts, but their actions are very high. So, what is required is recycling. So, cholesterol, what happens, it forms bile acid. These bile acids are stored in gallbladder and is released in intestine. Now, in intestine, this undergoes reabsorption. So, reabsorption around 12 to 32 grams per day are bile salts are reabsorbed and returned to liver for recycling. So, there are around 95% of efficiency, and the recycling is for around 6 to 8 times per day. Now, these absorptions are basically in the intestine, where active absorption occurs in ileum, whereas passive absorption occurs in jejunum. So, you can see if only this 0.2 to 0.6 grams of per day bile acids is synthesized in liver, but they are constantly recycling around 6 to 8 times, and what is actually excreted through feces is 0.2 to 0.6 grams per day. This is our loss, which is actually synthesized here. So, this is the efficient bile salts enterohepatic circulation. So, these are the points which we have discussed today, that cholesterol synthesis is mainly occurring from the precursor unit acetyl coenzyme following a complex series of reaction, and this synthesis is mostly regulated by hormonal control as well as intracellular concentration of cholesterol. So, this is all for today. These are my references. Thank you all. In the next class.